Well, welcome to the Department of Medicine Grand Rounds today here in the middle of March the 14th. And I'm excited to introduce our speaker today, who is someone who is relatively recently arrived to Emory, but there has been a lot of excitement and buzz around your arrival. And so I'm thrilled to actually get to both meet you and hear you talk. So today I'd like to introduce Dr. Anant Madabhushi, who joined Emory in February 2022 from Case Western Case Western Reserve University, where he came already as an established global leader in developing artificial intelligence to improve outcomes for individuals with cancer and other diseases. And also has used this technology to address health disparities as well, which is something very near and dear to many of us. You here at Emory, he holds the Robert W. Woodruff Professor of Biomedical Engineering chair and is on faculty in the departments of Pathology, Biomedical Informatics, and Radiology and Imaging Sciences. He's also a research career scientist at the Atlanta VA. Dr. Madabhushi has authored more than 475 peer-reviewed publications and more than 100 patents either issued or pending in the areas of artificial intelligence, radiomics, medical image analysis, computer-aided diagnosis, and computer vision. He is a fellow of the American Institute of Medical and Biological Engineering and of the Institute for Electrical and Electronic Engineers, as well as the National Academy of Inventors. In 2017, he received the IEEE, that's the Institute for Electrical and Electronic Engineers, Engineering and Medicine and Biology Society Award for technical achievements in computational imaging and digital pathology. His work on smart imaging computers for identifying lung cancer patients who need chemo was called up by Prevention Magazine as one of the top 10 medical breakthroughs in 2018. In 2019, no less than Nature hailed him as one of 5 scientists developing offbeat and innovative approaches for cancer research. And he has been named in the pathologist's power list of 100 inspirational and influential professionals in pathology the last 4 years running. So I'm delighted to introduce you, Dr. Madabhushi, to give Medicine Grand Rounds today. The title of your talk is Interpreter of Maladies, Applications of AI to Multiple Problems in Precision Medicine, or actually as written there. So thanks so much for joining us, and I will turn it over to you.
Great. Thank you. Thank you very much, Wendy, for that very kind introduction. I must warn everyone that after that very flattering introduction, it's probably going to be all downhill from here on. But just one clarification. I actually joined a little later than Feb 2022. I actually joined July of last year. So tomorrow is my 8-month anniversary. So just still in the process of settling in. Want to talk today about the work that our group's been doing, not just in oncology, but covering some other diseases as well. I want to talk about some of the work we're doing in diabetes, cardiovascular disease, as well as kidney disease. First, just want to address some disclosures. Want to mention that I have co-founded a couple of companies. One of these, Picture Health, is a company that has licensed some of the IP that I will be talking about today. I also have a number of sponsored research partnerships with pharmaceutical companies, and also have consulting arrangements with some companies. I will probably, though, say that my number one disclosure is the fact that I do have a social media account. And that's my Twitter handle. If any of you are on Twitter and any of what I present today is remotely appealing, please feel free to give me a follow. I guess I should also add that my second follow-on disclosure is the fact that none of what I'm going to talk about today was generated by ChatGPT. All right. So I want to set the stage a little bit for AI and computational imaging. And there's been a lot of discussion over the last 5 to 6 years in particular around the role of AI from a disease diagnostic perspective, that is being able to identify the presence or absence of disease from routinely acquired data. I would argue, however, that there is potentially an even bigger opportunity to use these tools to be able to prognosticate disease outcome, to be able to prognosticate disease progression, and critically, from a clinical standpoint, being able to predict response to treatment. And in my humble opinion, it's really the development of AI tools to develop both prognostic and predictive tools that will ultimately help realize the true promise of precision medicine, which is allowing us to tailor therapy for a given patient based on their specific risk profile. One of the things that we are seeing in clinical medicine is the fact that fairly routinely, we're acquiring multiple different streams of data. We have data that is being collected across multiple different length scales. And let's use here as an example, prostate cancer. What you're seeing is a region of adenocarcinoma on an MRI scan. And this is a T2-weighted MRI scan that captures morphologic attributes of the disease. Now, it turns out that with an MRI, one can actually acquire multiple different parameters. One can look at diffusion-weighted imaging. One can look at spectroscopic parameters associated with the tumor. One can look at vascular features through a dynamic contrast and enhanced MRI scan. Now, assuming this individual goes on to get a radical prostatectomy, we can actually take the specimen, digitize it with a whole slide scanner. And now with that high resolution digital image, we can start to apply AI and machine vision tools to start to interrogate the appearance of the tumor morphology in a highly quantitative way. So we can start to look at the spatial architecture and arrangement of individual cells, but we can start to look at the shapes of individual cancer cells. And then, as is routinely happening, we're also acquiring some kind of genomic or molecular level information for these diseases. And one can look at that same area that I identified on the MRI scan, apply things like laser capture, or more and more, we're seeing things like single cell or spatial transcriptomics to also be able to extricate a molecular characterization or molecular descriptor of that tumor. So we've got information that's really spanning the radiologic length scale to the pathologic length scale down to the molecular or omic length scale. And this really provides an unprecedented opportunity for us to think about AI from a knowledge alignment perspective. How can we start to align these various disparate sources of information? How can we start to extract novel features and novel measurements of the disease across these different length scales? And then how can we start to represent and combine the plurality of information that resides across these different length scales so that we can start to think about creating integrated predictors for improved diagnosis, prognosis, as well as treatment response. Now, today, when we talk about AI, it usually is conflated with a particular flavor of artificial intelligence called deep learning. Now, deep learning has emerged as a very powerful AI algorithm or class of algorithms for AI that has allowed us to do some very, very clever things from a wide variety of different applications. And around 6 or 7 years ago, these algorithms started to be applied to medical imaging data. This is, in fact, some of our own work published almost 9 years ago, one of the first instances of deep learning being applied to pathology images to be able to identify individual cancer cells. Now, the fundamental premise of a deep learning algorithm is that it uses what are called neural networks. Now, the concept of neural networks, of course, have been around for a long time. The main idea is you've got a series of what are called neuronal structures. These neurons essentially represent the information using sort of subsets of the input feature vector. And then using different ways of connecting these neurons, you have different architectures. So depending on the particular configuration, the particular architecture of the deep learning, you have a series of different types of neural networks that one can stand up. But at its core, or at its base, all of these deep learning algorithms are predicated on the fact that if you've got annotated data, you can then provide it to the AI, and the AI attempts to learn information or representations in particular that allow it to distinguish the target of interest from everything else. So in this particular case, we provided annotations of the cells of interest, specifically cancer cells, to the network, and the network then learned representations to distinguish the cells from everything else. And so you could see here that the only thing that was really required were annotations of the cells of interest to the network. The network was then able to go in and identify these target cells on new images. And that's really powerful because it means that without really having a deep knowledge or appreciation of the domain of this specific problem, we can stand up networks fairly efficiently and can use it to now go in and find targets of interest. But therein lies the rub. The challenge is that we don't really understand what these representations mean. We don't understand what the network is learning. And that's why a lot of these approaches have been labeled as mysterious machines. They're often called black box approaches because we don't really have an understanding of what the network is learning, and therefore there is this opacity with regard to these representations. And I want to give a specific example of where this black box approach really could land one in a catastrophic failure mode. This is work that we published, I think about 5 or 6 years ago. We were working with the University of Pennsylvania on a problem using machine learning to look at endomyocardial biopsies to be able to identify the presence of heart failure. This is with Ken Margelis and Elliot Paster and others at the University of Pennsylvania. They sent us 100 biopsies, endomyocardial biopsies of patients. And there was a mix of patients both with presence of heart failure on the endomyocardial biopsies, as well as patients without any evidence of failure. We trained a deep learning network, specifically a convolutional neural network, to try to distinguish between the presence or absence of heart failure. And in this case, this was what one would call a truly unsupervised approach. And in other words, the algorithm just ingested all the data. It had labels at a slide level, so we knew whether a patient had heart failure or not. That was all the information that was provided to the network. The network then learned representations to try to distinguish heart failure from not. We then took that network or that algorithm and then applied it to an independent set of another 100 images, also from the University of Pennsylvania. And just for laughs and giggles, what we also did was we compared the performance of the network against 3 expert pathologists, and really had the pathologists look at the same images in the test set and come up with a prediction of whether there was heart failure or not. When we looked at the results on the test set, we were surprised to find that the network comprehensively beat out the pathologists by about a 24% differential. The pathologists had an average area under the curve of about 0.73, 0.74. The network came back with an area under the curve of 0.97. So we were ecstatic. We went out and did a press release. GE Reports picked up the story and ran with it as one of the most exciting things happening on the planet that week. I was briefly trending on Reddit for a while, so it was all hunky-dory. But what happened about a month later was we got another tranche of images from the University of Pennsylvania, so same lab, same scanner that was used to digitize these images. And we took that same tranche of images, and ran it through the same algorithm that we previously constructed, and found, interestingly, that the network that had been at those dizzying heights of 0.97 in terms of AUC now fell precipitously down to 0.75. It's essentially coming down to the level of the pathologist. And we, for the longest time, could not understand what had happened, because we were talking about images from the same demographics, same population, same lab, same scanner. So what had happened? Ultimately, my students, who are way smarter than I am, were able to figure out that a remote software upgrade had been applied to the scanner between the first tranche of images and the second tranche of images. And that remote software upgrade had very subtly changed the appearance of the images. So even from from a human visual perspective, there was really no difference in the appearance of the images. The change was subtle enough to perturb the networks, and that's why the network had gone from that 97% down to 75%. And that sort of really segues into the main premise of this presentation, which is that we need to be wary about trusting these brute force algorithms too much, and really trusting these too much really is a faith misplaced. In fact, the example I like to use here is one that came out of UC Irvine, where Samir Singh, a professor of computer science, with his student trained a neural network to distinguish huskies from wolves. And as I'm sure all of you appreciate, this is a really difficult problem. They're very similar looking animals. But the network came back with an accuracy of 99%. Now, Samir, who's clearly way smarter than I am, because when he got those results, he did not run to his media communication office and say, I want to do a press release. He actually went back and tried to study what was going on. And what they found, which was really interesting, was the fact that the network recognized the fact that images of huskies invariably had a white background, right? Because huskies tend to favor colder climes, and there was snow in the background. That's what the network was picking up. It wasn't actually any features of the face of the animal. It was what was happening in the background. And so this is a fundamental issue, because when we don't understand the representations, when you've got a black box, then you really have to be very wary. And Cynthia Rudin has spoken very eloquently about this. Cynthia is a mathematician and computer scientist at Duke, and she's really talked very eloquently about the fact that these black box models, particularly for high stakes decisions like in clinical medicine, need to be approached with a great deal of caution, because if we don't, then that could really cause catastrophic harm because of the fact that there is a lack of interpretability. And she talks about how we really need to design models that are inherently interpretable. And so that really builds into the key thrust of my talk, which is really talking about some of the considerations for AI in the context of precision medicine. And I want to touch on three different topics. One is this issue of interpretability that I've already introduced. I'll talk about sort of black box approaches versus intentionally interpretable approaches, and how do you construct intentionally interpretable or engineered approaches. I want to talk about the issue of reproducibility, again, directly inspired by our catastrophic failure looking at endomyocardial biopsies, and the need to really look at the utility of these approaches across a plurality of sites. And finally, I want to talk about the issue of equity, because as we talk about these AI algorithms, we have to be intentional about the fact that they do need to work across a diversity of populations. And we've got to make sure that it's not only one particular population that these tools might be working in. And so what I'd like to do is to really go through these different considerations and pepper you with some examples of work going on in our group. And I want to start by talking about this issue of model interpretability. Now, I want to also quickly mention that, I'm not suggesting that we completely dismiss deep learning algorithms. Now, yes, they are black box approaches, but they are powerful and they are, can be very accurate. And so what we have really landed on are approaches in which we can try to blend the power of these very powerful deep learning approaches, but then use it with more interpretable, engineered feature representation approaches. So here's an example in the context of radiology where we're looking at chest CT scans, where we can leverage the power of deep learning to first identify the tumor, the vessels, the parenchyma, but then having done a detailed quantitative partitioning of the different compartments on a CT scan, we're now able to extract more interpretable features, such as features relating to the heterogeneity of the tumor, the peritumoral region, the shape of the tumor, as well as the shape of the tumor-associated vasculature. Similarly, we can develop similar strategies for pathology, where we can leverage the power of deep learning to go in and compartmentalize the different regions on an image, identify the different cellular compartments, the different tissue compartments, the stroma, the epithelium, but then start to pull out features that relate to the architectural arrangement and the interplay of cancer cells, stromal cells, tumor-infiltrating lymphocytes, and convert the pathology image into a series of quantitative digital biomarkers. And what we've actually shown over the last half a dozen years is that across multiple different indications, that these deep phenotyping approaches, this combination of black box, but with engineered features and more intentionally interpretable approaches, can allow us to predict treatment response across a whole plurality of different cancer types. And you can see here to the left, multiple different cancer types, in the middle, different combination therapy strategies, and then individual combination therapies where we have brought these approaches to bear both across radiology and pathology to be able to predict treatment response. So I want to go into some specific examples across multiple different domains where these approaches can be brought to bear for addressing questions in precision medicine. And I want to start off with work we're doing in the context of kidney biopsies, of kidney pathology. This is benign kidney disease, such as FSGS and CKD, where we're using the power of deep learning to do a deep phenotyping and compartmentalization of the different primitives on kidney biopsies. So we're able to use these powerful approaches to segment out glomeruli, to segment out proximal and distal tubules, to identify interstitial capillaries and vessels. And like I showed before, these deep learning algorithms can be very powerful if they are used in an appropriate way. In this case, we were using them for segmenting and partitioning out the different structures on these images. But having done that, it now provides an opportunity to now extract more interpretable features and trying to look at the association of those interpretable features with outcome. Specifically, in a paper that is in press and should be out next week in Kidney 360, we showed that looking at the features of peritubular capillaries, segmented first by deep learning, but then looking at shape-related attributes, so just looking at the major minor axis of the peritubular capillaries and looking at the shape, we were able to find a strong association with outcome in these patients with kidney disease, specifically looking at 40% eGFR decline. And the beauty was we simply looked at the shape of these peritubular capillaries. All of them had been segmented out on these kidney biopsies by deep learning, and then pulling the features out, we were able to see a very nice, strong association with one feature, with just one feature between patients who were likely to have a 40% eGFR decline or kidney failure versus patients who were not. Now, that's one example. Here's another one. This is in the context of breast cancer, where one of the big questions in early stage breast cancer is trying to figure out which women with early stage breast cancer are going to respond to chemotherapy or benefit from chemotherapy versus those women that are not going to benefit from chemotherapy. And one of the things that we know about early stage breast cancer is that the vast majority of women with early stage breast cancer will actually not require adjuvant chemotherapy. They'll do fine with hormonal therapy and surgical intervention alone. And now, about 20 years ago, Genomic Health, now part of Exact Sciences, came up with a 21 gene expression-based assay called Oncotype DX. And the beauty of Oncotype DX was that it provided a risk score where that risk score could now be used to identify which women needed the chemotherapy versus which women did not. There were challenges with this test. One was that it was tissue destructive. Two, it was expensive. It cost almost $4,000 per patient. But also, because it depended on analyzing a specific portion of the tumor, it was suspect in terms of the ability to capture the heterogeneity of the disease. And so our group, over the last decade or so, has been looking at trying to find ways in which we can interrogate routine H&E images and find interpretable features from these H&E images that can provide predictions of outcome and treatment benefit or chemo benefit, but doing it in a completely non-destructive way, doing it just solely of H&E images. But one of the things that we've been very focused on, like I said, is interpretability. And in this paper that we published about a year and a half ago in NPJ Breast Cancer, we looked at collagen. And specifically, the idea here was to try to identify with machine learning the collagen strands and collagen fibers on H&E images. So we were able to do this quite successfully just by using deep learning. Having done that, we were able to identify the tumor nests and then looked at the orientation of the collagen fibers with respect to the tumor nests. And what we found was really interesting, that when you looked at the relative organization of the collagen fibers with respect to the tumor nests, what we found was that in patients who had short-term survival, there was a lot more chaos with regard to, or rather, there was a lot more coherence, a lot more organization with regard to the collagen fiber architecture in patients who tended to do poorly. On the other hand, if you looked at patients who tended to have much better survival, the collagen architecture was more chaotic, was more disordered. And so we actually used an idea from physics, the concept of entropy, to be able to capture the relative order or disorder with regard to the collagen fibers on the H&E images. And what we found was that the single entropy feature, looking at the collagen orientation, was able to stratify patients in terms of their survival outcomes. And so we did a study with clinical trial data sets as well as patients from the TCGA or the Cancer Genome Atlas, and showed quite nicely that this single feature, just collagen fiber architecture, lack of orientation or disorder feature, was able to stratify outcomes. In a subsequent paper that is currently in press, also in Nature Breast Cancer, we did a follow-on study where we looked specifically at the high and the low Oncotype DX categories. So remember, the Oncotype is a molecular-based test, the $4,000 test that is essentially providing a score where the low score suggests women perhaps may not need the chemotherapy, and the high score suggests that women may actually benefit from the chemotherapy. When we looked at women who actually got the low or had a low oncotype DX risk categorization, what was really interesting was we found that the image classifier, the image features, were able to very nicely stratify outcomes within the low oncotype DX category. So in other words, we were able to identify a group of women who did very well, but then also identify the subset of women who are going to do poorly. Now remember, low oncotype suggests that these women should not be getting chemotherapy, but our data is showing clearly that there's a subset of women, even though they had a low oncotype DX score, that they probably were going to benefit from adjuvant chemotherapy. Conversely, if you look at the high oncotype DX risk category, what we're finding is that there is a subset of women identified by the image classifier who are likely going to have much better, more favorable prognosis, suggesting that in hindsight, these women could actually have avoided adjuvant chemotherapy. So what you're seeing is a potential of these tools to not just have a prognostic benefit, but also a predictive benefit. So talking about a predictive benefit, here's an example of work that was published last year in Science Advances, where we were looking at the spatial architecture or the spatial arrangement of tumor-infiltrating lymphocytes. Now, there's been a lot of work around the role of immune cells or lymphocytes on cancer biopsies and quantifying the number of lymphocytes and finding an association with outcome and response in many different indications, cancer indications. We did something slightly different. Instead of looking just at the number of lymphocytes, we focused instead on architecture and specifically trying to understand with AI, with network theory, trying to quantify the spatial statistics relating to the interplay between cancer cells and tumor-infiltrating lymphocytes on H&E images. And we looked specifically at patients with non-small cell lung cancer that had been treated with immunotherapy. And we found that when you quantified the spatial relationships of these two different cell types on biopsies in these lung cancer patients, that we were able to, one, predict response. We were able to predict objective response, whether these patients were going to respond to immunotherapy or not. But then B, we were also able to show a strong association with overall survival in patients across multiple different sites and also across multiple different agents. We were able to look at atezolizumab, pembrolizumab, as well as nivolumab, and showed across 3 different immunotherapy drugs that we could see a strong association with outcome. And perhaps best of all, the data, this was, in my mind, the most exciting data that we showed in the paper, which is looking at patients with low PD-L1. Now, of course, I'm sure all of you are aware that PD-L1 is the biomarker that's currently used for identifying patients who are going to benefit from immunotherapy or not. But it's well known that PD-L1 is not a very good biomarker. And patients who typically have low PD-L1 either are not recommended for immunotherapy or are likely to get a combination of immunotherapy and chemotherapy. When we looked at patients specifically with low PD-L1, we found this tumor-infiltrating lymphocytic signature allowed us to distinguish between patients who had a much more favorable risk profile versus patients who had a much more unfavorable risk profile. And why that is exciting is because it suggests that for patients that land on the yellow curve, that these are the patients who actually might benefit from mono-IO or mono-immunotherapy as opposed to a combination of immunotherapy and chemotherapy. So we came back to the problem that I started out with, which was looking at heart failure. And this is work that we actually published in the European Heart Journal last year. So we went back to the same problem, but this time, instead of using a black box approach, we went to look at these biopsies with a combination of the interpretable features that I just presented on the lung cancer cases, where we looked at the interplay or the interaction between lymphocytes and cardiomyocytes on endomyocardial biopsies, and found that when we looked at almost 2,300 biopsies across 5 or 6 different sites, that 2 things happened. One was we got very, very stable predictions across multiple different sites. But B, we actually ended up with a prediction that was higher than what we got with the black box approach. We actually got an area under the curve of 0.98 in distinguishing patients who were at high risk for rejection versus patients who were at a low risk for rejection. So this was exciting because it really brought that story full circle we learned that the black box approach could not really be trusted as an end-to-end classification paradigm. Going from there, taking the lessons we learned and applying it to developing a set of features that were intentionally interpretable, and then validating that not only did this approach do better, it also was resilient and robust across a larger cohort of cases across multiple sites, multiple labs, multiple scanners. We've continued to look at this particular motif, the lymphocytic infiltration motif across other use cases. This is work that we're just about to submit for publication, looking at ductal carcinoma in situ or stage 0 breast cancer, where we've shown that the spatial arrangement of the lymphocytes on tissue images of BCIS is not only prognostic of outcome, it is predictive of benefit of radiation therapy. So DCIS, there's of course a standing conundrum in the management of DCIS patients in that it's unclear as to which patients truly are going to receive benefit from adjuvant radiation therapy, whereas which patients perhaps might just benefit from surgical resection alone. And our data is showing that this spatial statistic feature relating to the lymphocytes actually identifies patients who are going to receive the added benefit of radiation therapy versus patients who might benefit from surgical intervention alone. So I want to switch over from talking about pathology to talking about radiology. And I want to start with this one example in the context of brain tumors. One of the big challenges in brain tumors is that these patients, after they're treated with chemotherapy and radiation, they come back for a follow-up MRI scan. And inevitably, something shows up on the MRI scan. And it's very difficult to tell as to what this signal is. Is it a recurrence of the tumor? Or is it radiation necrosis, which is just an effect, a benign effect of the chemoradiation therapy? And because we're unable to tell, very often these patients will be subjected to an intervention. They might be subjected to an intracranial biopsy to be able to prize out the tissue. What we decided to do was to see if we could develop a non-invasive way of interrogating the radiologic images of the MRI scans so that potentially we could tell without an intervention whether what we were seeing on the MRI scan was a tumor recurrence or radiation necrosis. And so similar to the collagen story, we actually looked at individual gradients or individual edges on a voxel by voxel basis and found that similar, again, to the collagen, we were able to capture the entropy associated with the directionality of these edges. So if you think about a pixel is surrounded by 8 other pixels, and you can compute the dominant edge of the center pixel with respect to the 8 other pixels around it. The arrows that you see here represent the dominant edge on a pixel by pixel basis. And then we're computing entropy, which again captures the disorder. And we're able to create color maps like the one that you see here, where the red, the deep red, essentially represents those areas with a high degree of entropy. In other words, those are the areas where there is significant morphologic heterogeneity. On the other hand, if you look at the right panel actually has significantly lower entropy, meaning that there is much more order, much more arrangement with regard to the underlying morphology. And it turns out that if you see this entropy pattern, it's really associated with the tumor recurrence. On the other hand, the lack of entropy is reflective of a more coherent microarchitecture, which in this case is associated with a benign effect or radiation necrosis. So we've taken this approach and applied it beyond brain tumors to multiple other indications. This is work we've been doing over the last 5 years with the Coulter Institute at the Cleveland Clinic, where we've been trying to find ways in which we can analyze both ultrawide fluorescein angiography images, but also looking at OCT scans of the eye to be able to characterize a variety of different diabetic conditions like diabetic retinopathy and diabetic macular edema. And we found that these radiomic patterns, these entropy patterns that capture the morphologic heterogeneity, actually tell us as to which patients are going to respond to anti-VEGF drugs. In this case, aflibercept, which is a drug to help alleviate diabetic macular edema. And we're finding that the extent of the entropy is directly correlated with the likelihood of response. In other words, if you have a great deal of heterogeneity, then you're likely to not respond to aflibercept, and the converse is true if the heterogeneity is low. And this is not just in the context of sort of diabetic diseases. You're seeing this in the context of neovascular AMD as well, where there's a strong association between anti-VEGF response and durability of response, again, with these radiomic features. Working with Dr. Meena Chung at the Cleveland Clinic over the last 6 years, we've been looking at the problem of atrial fibrillation and specifically trying to understand whether from routine CAT scans, we can identify features, we can identify patterns that might suggest which patients are at risk of recurrence of AFib post-ablation. And this was a project that started about 6 years ago. It culminated in an R01 that we were awarded a couple of years ago from NHLBI. And what we've been trying to do in this particular case is twofold. One is trying to develop predictors of recurrence and using AI to do this, but also more critically, trying to understand whether there are ways with the AI that we could predict what the sites of recurrence might be, because that's another unknown. Where in the heart are you likely to see the recurrence of AFib? And so here we use something called an atlas model-based approach, or a statistical model-based approach. And I'll sort of distill it down and sort of give you the punchline. Basically, we took a cohort of patients who did have recurrence of AFib. We looked at another cohort of patients who did not have a recurrence of AFib after ablation. We created a statistical model or an atlas model for all the patients who did have recurrence, and similarly created one for those patients who did not have recurrence. Then we did a difference, right? And essentially what the difference atlas showed us was that there were specific regions between these two populations that were distended. And what we found that was really interesting was that in the left atrium, the left atrial appendage, as well as in the pulmonary veins, there were significant shape distensions and shape differences between patients who did and did not have recurrence, suggesting that these areas might be implicated as sites of recurrence. What we've subsequently done is to look at a series of different radiomic features, again, these entropy features, but also looking at shape features of these particular areas that were found to be different in this statistical model-based approach. So we looked at shape and entropy features of the left atrium, the left atrial appendage, as well as the pulmonary veins, and showed that these features allowed us to be able to predict risk of recurrence after ablation with a fairly high degree of accuracy. So it's twofold. One is we're able to predict recurrence at a patient level, but it also suggests that these sites of recurrence that we've identified are candidate sites for preemptive ablation to mitigate the risk of post-therapy recurrence. This is work that we just recently published. This just came out in Lancet Biomedicine. This was led by Dr. Gaurav Modanwal, who is a research scientist in our group. And this is work that we've been conducting with Dr. Sanjay Rajagopalan and Dr. Sudhir Alkindi at the University Hospitals in Cleveland, where we looked at a series of patients with abdominal CTs and essentially used machine learning to quantify the extent of hepatic fat, and showed that the machine learning derived measures of hepatic fat were actually strongly associated with the severity of COVID-19. And we showed in this publication on a few thousand patients that there was a very strong association between the extent of hepatic fat to the severity of COVID-19. What was nice was that we had data from 3 different continents, North America, Europe, as well as Asia. And when we did the pooled analysis across multiple different populations, we found that these trends held up. What we've also done more recently, and this is a paper that we'll be submitting for publication very soon, is validated this machine learning based quantification of hepatic fat using MR spectroscopy. So MR spectroscopy is the gold standard for capturing the extent of liver fat. But what we were able to demonstrate was a very strong tight association between our machine learning derived quantification of hepatic fat from the CT scans with the measures that we were getting from spectroscopy. So building off this data, we have just completed a study that I wanted to share with you. This is still early, but we're really excited about this. What we've actually done now is trying to understand whether the heterogeneity patterns of the liver fat associate with downstream major adverse cardiovascular events. In one of the largest studies that we're aware of that we just conducted, we looked at 45,000 patients and across multiple different indications. And across these 45,000 patients, what we found is that there is a very, very strong association between the heterogeneity of the liver fat with downstream cardiovascular MACE events. Now, the heterogeneity or the entropy features are in a univariate fashion associated with MACE, but interestingly, when you combine the imaging features with a series of different clinical parameters and clinical factors, we're finding that the AUC goes up quite significantly. And you know, there's a pretty good AUC here, but what is perhaps even more interesting is a strong association with downstream MACE events. You can see here a lot of data, so I apologize, but across the board, what we're finding is that the imaging parameters alone are strongly associated with cardiovascular downstream MACE events, but then the combination gives us an even stronger signal. So in other words, combining the imaging and the clinical parameters, that imaging clinical nomogram actually gives us even stronger indication of which patients are likely to have downstream cardiovascular MACE events. In a paper that we just submitted last week, in fact, we've been looking at patients, CKD patients, chronic kidney disease patients, and looking at their echocardiograms. And Rohan Damdare, who's a grad student in our group, developed a machine learning model to be able to capture in a spatiotemporal fashion the dynamics of the ventricular wall. And specifically, he was able to show that when you look at the left ventricular wall and looked at the deformations in that spatiotemporal profile, that those features, the distension of the left ventricular wall was associated with downstream MACE. And so this is a relatively small study, 111 patients, but we're showing that we can tease out some really important information, even from echo, which of course we know is somewhat more difficult to interpret. But you know, it's very heartening to see that even from an echo, we're able to find an association with MACE outcome. COVID-19, we did another study last year, where we looked at patients who had both severe COVID, mild COVID, as well as patients who had no evidence of COVID. Put together about 4,000 CT scans across these different populations and created a statistical model. So we looked at a statistical model, or constructed a statistical model for patients with severe COVID, created one for patients with mild COVID, and then one for controls. And when we looked at the different atlases, what we found was clear evidence that there was significant deformation that was occurring in the lungs on account of more severe COVID-19. And this data we found typically tended to occur in the mediastinal surfaces, where there was a strong difference between the statistical models for the controls versus the mild, and the mild versus severe COVID-19 patients. Another intentionally interpretable feature that we've been looking at is with regard to the airway. So this is from routine CT scans. We're able to go in and use machine learning to tease out and segment out the airway. And finding that looking at parameters relating to the shape and a series of different statistics relating to the airway, the primary branches, secondary branches, tertiary branches, that we can start to look at multiple different pulmonary conditions. We've actually shown that we can distinguish severe COVID from mild COVID based off these features of the airway, but also, in fact, interestingly finding an association with response to immunotherapy. Just sort of keeping in line with this idea of interpretable features, here is yet another interpretable feature that we developed in our group. This was work that was published in Clinical Cancer Research last year, where we were looking at the tortuosity or the twistedness of the tumor-associated vasculature. And we actually found that if you looked at patients with breast cancer who were undergoing neoadjuvant anthracycline therapy, that the twistedness of the tumor-associated vasculature was implicated in a pathologic complete response. In other words, if the vasculature was smooth, those patients were likely to have complete pathologic response. On the other hand, if the twistedness of the vasculature was extreme, then those patients were likely to not respond to neoadjuvant anthracycline therapy and therefore were likely to not have PCR. We've shown that this feature is also implicated in predicting response to immunotherapy. But again, it extends it. In my mind, vascular tortuosity is really a pan-disease condition. This is work that we've been doing in the context of DRE and DME and diabetic retinopathy and diabetic macular edema, where we're looking at tortuosity of vessels in the eye on ultrawide fluorescein angiography images. And in data that we've generated in a few different publications, we've shown over and over again the twistedness of the vasculature in the eye is strongly associated with response to aflibercept, this anti-VEGF therapy for DRE and DME. So I've talked about imaging, talked about pathology. Well, what about 12-lead ECG? What can we do with 12-lead ECG? This is some work we published a couple of years ago in Circ AE, where we looked at 12-lead ECG in the context of patients undergoing cardiac resynchronization therapy or CRT and showed that by applying very simple principal component analysis and clustering analysis just to 12-lead ECG, we were able to stratify patients in terms of survival, patients who had undergone cardiac resynchronization therapy. So this was a total cohort of about 1,000 patients. We had about 540 patients in the validation set and showed that there was a very nice distinction between groups who had more favorable outcome from cardiac resynchronization therapy versus patients who did not have a favorable outcome. And this is important because only 20 to 50% of patients actually tend, only about 50 to 70% of patients actually do benefit from CRT, and somewhere like 20 to 50% of patients do not actually improve with therapy. So being able to identify that upfront and early, I think, would be beneficial in managing strategies for these patients. And another example of the use of ECG data, this was published just 3 months ago in Frontiers in Cardiovascular Medicine, where we showed again that machine learning with 12-lead ECG data coupled with coronary artery calcium scoring allowed us to prognosticate downstream MACE events in these patients, and a very, very strong signal emerging from the machine learning derived features of the ECG coupled with the coronary artery calcium score. And so finally, I want to end by talking about equitable AI. One of the reasons why we've been very focused on this particular problem is because of data like this. I talked about the Oncotype DX score, which has been used for identifying which women with breast cancer will benefit from chemotherapy. Unfortunately, this paper came out in 2021, a couple of years ago, showing that the Oncotype DX breast recurrence score actually did not do very well in Black women. And if you think about it, it really means that when this test was developed, there probably weren't a large number of Black women that were represented in the training set for developing the original test. And that's a problem. That is an issue because we're seeing a lot of these risk calculators not working across a diversity of populations. And that's exactly why our group has been very intentionally focused on trying to address this issue up front. This is work we published a couple of years ago in Clinical Cancer Research, where we looked in a very systematic way at tissue images of prostate cancer between Black men and white men, and identified that there were significant differences in the stromal morphology, particularly for Black men, where there was a lot more heterogeneity in the morphology of the stroma in Black men with prostate cancer. We then use these differences to create population-tailored models and actually showed that a population-tailored model for Black men allowed us to better prognosticate risk of recurrence compared to a population-agnostic model. In other words, if you didn't account for the population differences, then that model did not actually work very well in Black men. And so I think we have to be intentional. We've got to use AI to first discover whether there are differences across populations, and if there are, we need to be able to use these differences to create more population-tailored models. And then I just want to sort of come back full circle this concept that I talked about right at the start, which is combining information. How can we start to combine the plurality of data that we have at our disposal for so many different indications and get the most value out of it? And so this is exactly what we've been doing, looking at combining radiology and pathology images. This is an example in the context of head and neck cancer, P16 positive head and neck cancers, where we've shown the combination of features and measurements, both from H&E as well as from CT scans, allows us to better prognosticate patients in terms of recurrence for oropharyngeal cancers compared to looking at radiology alone or looking at the pathology alone. Of course, this has implications above and beyond head and neck cancer to other cancers, but also other diseases as well. So let me conclude. So hopefully what I've conveyed is that computational analytics can, with routine imaging data, can help address questions in precision medicine, specifically around prognosis and treatment response. Want to emphasize the low-cost aspect of these approaches, the fact that we're working with routine CT scans, ECG data, pathology data, data that is routinely acquired. And as an immigrant myself, I think about global impact. I think about countries like my own native India, where these tools can have an impact, countries that are low and middle income, where you don't have access for a $4,000 test. And so you really have to think about frugal technologies. But I also want to convey, hopefully, that with artificial intelligence, we have to be thinking about interpretability. We have to figure out ways in which we can establish a morphologic or molecular basis of the AI, because an intuitive basis is really going to be important to drive clinical adoption. Finally, just want to do a quick shout out. To the amazing students, trainees, postdocs, faculty, and scientists in my group. I just come in and try to take credit for their work. I mean, this is all them. They do all the heavy lifting. And I'm just so privileged and honored to be able to work with them, and so privileged and honored that a number of them agreed to move with me from Cleveland to Atlanta. And of course, you have to thank the people who put food on the table. So we're very, very privileged for the funding from the NCI, NIH, NHLBI, DOD, and other foundations. Thank you very much.
Thank you so much. That's frankly a little bit mind-blowing. And I have tons of things I could ask you, but Dr. Stevens, I see your hand up.
No, thank you very much. That was great. And as Wendy said, a very informative and engaging presentation. Thank you. I guess I have two questions. One is about the comment about combining modalities and extending those modalities to biologics and our biomarkers and genetics, and your comments about how to do that and how to do that effectively. That seems to be a significant challenge. And again, going back to the warning you presented initially about black boxes and how those can be deceiving. Where's the field? What do you think the prediction is for AI in medicine in the next decade to 25 years? So your comments to those. Thank you.
Great. Thank you, David. Excellent questions. I'm going to try to respond in about 30 seconds. So clearly I won't do justice to those very profound questions. I think the issue of combination of data is going to be really critical in my humble estimation. I think we've been leaving too much data on the table for too long, and we have to figure out ways in which we can integrate the information. Now, I can show data from the research perspective, but I think what your question really gets at is implementation. There are challenges there. We're talking about radiology, pathology. And I know that we've struggled even from a research perspective to be able to reconcile a CT scan with a pathology image. And so being able to implement multimodal diagnostic techniques is definitely going to be a challenge from an implementation perspective, because it means that the different streams of data have to be able to coalesce together for us to be able to then bring AI to bear on that. So a lot of that will have to do with figuring out workflows and figuring out how to be able to stream that data and centralize the data so that the AI can do its thing on that combined data. The second question around, sort of the me looking into my crystal ball I don't have a crystal ball, but I will say that again, my own bias or feeling is that ultimately you will have it's ultimately going to be driven by the consumer, right? And the consumer is all of you. And I think that the companies that develop their own flavor of AI are going to have to be able to present data that is going to be compelling, that's going to have to be validated across a plurality of sites and across multiple different populations. You know, that is the standard that I think these companies and vendors are going to have to pass if they want to bring their products to market. I see that the regulatory agencies are going to become more vigilant about this. I think that for some time, a lot of these companies have had a bit of a free pass in the sense that the validation studies with a lot of the AI approaches have been really meager. I read somewhere that the average validation set size for some of these AI tools for a 510K submission is about 300 patients, which to me feels woefully inadequate when you consider the fact that we're seeing potential differences across populations. I don't think a lot of these companies are really presenting data across a diversity of populations. And so just as I look into the future, I think we have to insist on interpretability. And more critically, we have to insist on the fact that these tools, the data that's being reported encompasses multiple different populations. We have to ensure from the get-go that there is equity that's built into these tools. So I think that the companies that do that right and the products that are able to speak to the interpretability, that are able to connect with the physicians and demonstrate the fact that they are working across different populations are the ones that are going to be winners. Carlos?
Thanks. Yeah, Carlos, and then Dr. Akbashev, and then I think that'll be it time.
Yeah, well, thank you, Dan. This was fantastic. And again, there's, as Wendy said, so many questions. But I think about, the amount of data that we generate on an annual basis nowadays, the amount of data that healthcare is generating. And I think about ourselves as physicians, but I also think about our students and trainees, and I feel like we may be way behind. So what would your advice, or your quick advice be for young trainees, people starting practice, about how do they get involved in this? How do they get to understand it? Because this is going to be how they're going to practice.
Yeah, so Carlos, I mean, that's a great discussion. I think something that I'm hoping that to talk to you and others on going forward. I think there's a big opportunity to continue to train, folks on the technical aspects of AI. But I think a lot of physicians, I see residents, I see medical students, I see attendings, reaching out saying, hey, I want to learn about AI. So I do think that as an institution, I'm sort of wearing my Emory/Grady hat on here and show as well. I think we have to think about ways in which we can make the AI more accessible. We've got to think about ways and constructs here organizationally that we democratize AI, because we want more people to be engaged. We want them to understand the opportunities. But really, I think the work that I'm showing to me is just a small fraction of what is possible. And I think if we do this right and we think about setting up the right infrastructure, we can then essentially make AI possible for a large number of physicians, specialists, residents across the gamut. I think that we want to train our residents, we want to train our students. But at the same time, I think there's an opportunity to engage attendings as well and get them to appreciate the AI so that we can start to think about really moving this, into implementation, because I think that's really where we need to go.
Thank you very much.
Mikhail?
Thank you. And my question is simple, and thank you for the interesting talk. It's what types of problems are machine learning the right method to address? And then when is it if I read a paper and somebody uses machine learning to figure out something, and when should I be skeptical and when should I be impressed?
Yeah, that's a great question. I think that you need to be skeptical when it's the husky and the wolf, right? When somebody tries to sell you the husky and the wolf, that you got an amazing algorithm that gives you 100% accuracy in distinguishing huskies from wolves. When you know that this is a really challenging problem for the human, and I think that's really where you need to be skeptical. Because one of the things, or the lessons that I've learned in this journey, in my AI journey, is that you really have to think about value add, and not to be too quick to criticize or disparage the vast knowledge bank that exists within clinical medicine, right? I think that very often there's a certain arrogance with AI and AI companies and AI vendors. You come in, yeah, we're going to teach physicians how to do their job. But what they don't appreciate is the vast amount of knowledge that has been accumulated over hundreds of years, really. And so, the reason I mentioned that story about the biopsies is because it's a very humbling story, because it was the realization that no, AI can be wrong, it will be wrong. And unless you truly appreciate, how the AI works, you're not going to be able to come up with a solution that you can really trust. So I think just as you look at publications, I think you want to be thinking about those opportunities where maybe there's a great deal of tedium involved, where it's a solvable problem for a machine, but where perhaps it just takes too long. When I talk about pathology, I always talk about mitotic figures. It's the classic problem that makes the pathologist's eyes bleed. They can do it, but man, that is, you talk about a problem that's apt for AI, that's one. So the thing that I would suggest is really in the context of your particular specialty, what are those low-hanging fruit where I think it's just so tedious for a human to do those things, but you could train an AI algorithm. Those, in my mind, represent the low-hanging fruit.
It's so interesting. And I think I wish we had like another hour for questions, because I think many of us have many more. And I've already thought of the examples in my own specialty, but thank you so much for joining us today. This was absolutely fantastic and really opens a window into the future that is exciting. Honestly, I think people often look at AI and think it's going to put doctors out of business, but I actually don't feel that way at all. Having listened to you speak, and really am enthusiastic about where how this could help us.
Wonderful. Thank you. Thank you for giving me the opportunity.
Thanks.
Cheers. Bye.
Bye.